Very good morning to Shibhadra ma'am and my all dear friends. Today I'm going to explain you all glycolysis pathway. So glycolysis pathway is known as the foundation pathway for both aerobic and anaerobic respiration in cellular beings. And this pathway is a 10 enzyme catalyzed reactions in which the starting molecule, which is glucose, is at last converted to pyruvate. So in the first step, the glucose is converted to glucose 6 phosphate and ATP is changed to ADP, and the enzyme catalyzing this reaction is known as hexokinase. And glucose 6 phosphate and glucose is all are 6 member glucose like ring structures. And the process and hexokinase is the enzyme which is catalyzing this reaction, and this is also known as phosphorylation process. So mainly the phosphorylation process is also known as adding of phosphate group to a molecule derived from ATP. So and the ATP is changed to ADP here, and it is the first step, first reaction of energy investing phase of glycolysis pathway. In the second step of glycolysis involves the conversion of glucose 6 phosphate to fructose 6 phosphate. The reaction occurs with the help of the enzyme phosphoglycoisomerase. And the reaction involves the rearrangement of carbon oxygen bond to transport the 6 membered ring into 5 membered ring to and the rearrangement takes place, and the 6 member ring opens and then closes in such a way that the first carbon becomes now the external to the ring. So in the third step we have of glycolysis, we see that fructose 6 phosphate is converted to fructose 1,6 bisphosphate and ATP is changed to ADP. And this process is irreversible in nature and similar to first step of glycolysis, because we see that in the first step glucose was changed to glucose 6 phosphate and ATP was changed to ADP. So here also fructose 6 phosphate is changed to fructose 1,6 bisphosphate and ATP is changed to ADP. Enzyme catalyzing this reaction is phosphofructokinase. So in the fourth step, the enzyme used is aldolase, which catalyzes the cleavage of fructose bisphosphate to yield 2 3 carbon molecules. One of these molecules is called glyceraldehyde 3 phosphate and other one is called dihydroxyacetone phosphate. In the fifth step, the enzyme is phosphate isomerase rapidly converts the molecules of dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. The glyceraldehyde phosphate is removed and used in the next step of the glycolysis. The glyceraldehyde phosphate is the only molecule that continues in the glycolytic pathway. As a result, dihydroxyacetone phosphate molecules produced are further acted on the enzyme triisophosphate isomerase. In the sixth step, we see that in the step glyceraldehyde 3 phosphate dehydrogenates and add an organic phosphate to glyceraldehyde 3 phosphate, producing 1,3 bisphosphate glycerate. So in this step, two main events take place. First is glyceraldehyde 3 phosphate is oxidized by the coenzyme and the molecule is phosphorylated by the addition of a free phosphate group. And the enzyme catalyzing this reaction is glyceraldehyde 3 phosphate dehydrogenase. In the seventh step, we see that 1,3 bis phosphoglycerate is converted to 3 phosphoglycerate by the enzyme phosphoglycerate kinase. The reaction involves the loss of phosphate group from starting material. The phosphate is transferred to a molecule of ADP that yields ATP, and with the synthesis of ATP we have the first two molecules are cancelled and the ATP is used, so there is no ATP left. So you can see in the reaction that there is no ATP left, and so we move on to eighth step, that is the enzyme phosphoglycerobutase. uh, and this step involves a simple adjustment of the position of the phosphate group. In this step only the phosphate group is repositioned, and making the 3 phosphoglycerate molecule to 2 phosphoglycerate molecule. In this only the position of phosphate group is changed, and this phosphate group is responsible for catalyzing this reaction is called phosphoglycerate mutase. A mutase is an enzyme that catalyzes the transfer of the functional group from one position to another position. So in this the enzyme which is transferring 3 phosphoglycerate molecule to 2 phosphoglycerate molecule is phosphoglycerate mutase. And then in the ninth step, this involves the conversion of 2 phosphoglycerate to phosphophenol pyruvate. The reaction is catalyzed by the enzyme enolase, and enolase works by removing a water group or dehydrating the 2 phosphoglycerate. The specificity of the enzyme pocket allows for the reaction to occur through a series of steps too. And in this mainly in this step, the water group is removed, and the 2 phosphoglycerate is converted into phosphophenol pyruvate. So 2 phosphoglycerate also have 2 molecules, and phosphophenol pyruvate also have 2 molecules. And in the last step, the enzyme pyruvate kinase transfers a phosphate from phosphophenol pyruvate to ADP uh, to form ATP. The final step of the glycolysis converts the phosphophenol pyruvate into pyruvate with the help of the enzyme pyruvate kinase, and then it finishes the glycolysis pathway, and then the cell continues the respiration either in the aerobic mode or in the anaerobic mode, and this choice is made on the circumstances of the particular cell. So here we end the glycolysis pathway, and I hope you all have understand. Thank you for listening.